Hello all, my name is Samir Almastri and I will be presenting to you my research titled Comparing Metabolites Within the Media of Mesenchymal Stromal Cell Donors During Expansion. So, to begin with, mesenchymal stromal cells, also known as MSCs, which I will be referencing as such throughout the presentation, have the capacity to possess tremendous potential to transform cell therapy as we know it today. So, as this diagram shows, a lot of that is due to their lineage differentiation because of their multipotency, meaning they can differentiate into different cells types. And then in addition, their expansion potential, so they have rapid proliferation and they're easy to grow, in addition to the fact that they can be grown in 2D cultures and 3D cultures. Thirdly is their ability to adhere to plastics, and this is planar plastic surfaces and carriers, which is very imperative to how we culture cells today. And then in addition, the transient Pacrian actions, which pertains to the secretion of different hormones and growth factors and signaling proteins and relevance to the end cell function of these cells. And then lastly is the modulatory properties that MSCs possess, and that pertains to immunosuppression and the inhibition of proliferation of invasive cells as far as treatment methodologies. The objective of this research project was to discover markers of the MSCs grown in culture that can be predictive of end cell function of cell lineage differentiation through investigation of the relationship between the extracted metabolites, which was done using NMR, in the alpha MEM media in relation to specific donors. Next, let's discuss the significance of the study. Firstly, the heterogeneity of MSCs. This attribute of MSCs makes it difficult to establish reproducible and consistent results. Secondly, it is difficult to obtain consistent and reproducible end functions of the cells due to the diversity among tissue source donors. Thirdly, optimal growing conditions are still yet to be discovered. And lastly, identification of predictive markers for end cell function during differentiation can serve to lower cost and risk potential for treatment discoveries. Let's now discuss the MSC expansion workflow that was conducted. So to begin with, 2 samples of each tissue donor source was taken and combined with either the Rooster biomedia or the alpha MEM media. From there, the combination was then inserted into a T75 tray, and 10 replicates of that were made, with 1 media control. From then, between those 10, they were all randomized for labeling to insert into NMR. The bulk media was then collected and replaced every 3 days, and that was labeled at different time points. In addition, it was all grown in standard cell culture conditions. And then lastly, the media collection was continued until plates resulted in 80% confluence of MSCs in each tray. So now let's look at what an NMR spectrometer is. NMR stands for nuclear magnetic resonance. This diagram shows how it works. So the sample is placed in a tube into the magnet. The magnetic field is strong enough to extract either the metabolites or the protons in order to gain a further understanding of the structure, identity, concentration, and behavior of the molecules, which in this case were within the media. With this, a 1.7 microliter sample was taken from each tray and placed into the NMR. NMR spectroscopy was then ran on each individual sample, and then the detector reported the absorption rate to the printer. So now to continue with the methods, what we began to do after obtaining the NMR spectra was select possible compounds from the NMR spectra using Colmar. We then performed comparative analysis by overlaying the compound spectra onto the alpha MEM NMR data. We then confirmed extracted metabolites with the toxi data using MNOVA. So here is an example of the image of data processing done with a lactic acid peak from the sample. On this report, we can see that at 1.3 and 22.9, the circled peak is shown here, and we can associate that with the triplet seen at the very top. This would then be classified as the lactic acid peak of the data. From the data, we were then able to configure a heat map which compared donors and metabolites. This heat map shows correlation between metabolites and donors. Here it is important to note the differentiation between donor metabolite correlation of the same donor. More specifically, donor RB174.2 and RB174.1 represent the same donor, yet the MSCs were plated and harvested at different times. This now highlights that depending on when the MSCs were plated and harvested, the metabolite intensities in compared to donor classifications can differ. Next, a one-way ANOVA test was ran. This represents the singular significant feature glucosamine reported by the one-way ANOVA test. Donors RB174.2, RB175, and the MSCs have the highest level of glucosamine seen by this test. This is significant as it can be a predictive of MSC integral output. We then created a heat map of the correlation of metabolites at time point 1. The significant metabolite categorized by MNOVA, which was glucosamines as we just saw previously, is closely related to the presence of aspartate, lysine, and methionine at this time point. This relationship can highlight predictive markers of the integral output of the cell. Next, we performed an IDO assay assessing MSC and modulatory functioning. So IDO assays were normally used as a corollary of MSC potency. They are performed to assess MSC and modulatory functions. On this graph, MSCs have the highest kynurenine production without consideration of certain outliers, which were removed from previous analysis. High kynurenine production suggests that the cell will perform well based on several previous studies. The conclusions we gained from this study were, first, MSCs can be classified as the best performing cells based on their overall consistency and kynurenine production. Secondly, despite being from the same donor, time of cell culture greatly affects production of various metabolites in media. And thirdly, glucosamine intensity at time point 1 in the media was indicated as a significant figure based on the ANOVA test and can be connected to the potential function of the cell based on statistical analysis. This then demonstrated favorability towards two specific donors RB174.2 at this time point and RB175, in addition to the MSCs. Now, for future directions, one of the next steps would be to compare the composition of the rooster biomedia and the alpha-M media at the different phases of harvesting to identify differentiation and potential predictive integral output of the cells. Next, the next step would be to also identify predictable relationships to metabolite excretion of MSCs with the function capabilities of each cell. So, moreover, how they differentiate based on certain predictive markers and the integrity of the cell and its function. Thirdly, it would be to classify the donors for the MSCs in connection to the culture media and donor-specific cell differentiation to then identify the optimal donor source, the optimal time point, and the optimal growing conditions of each cell to ensure optimal cell end functioning. And last but not least, here is my acknowledgement page.